Aging means cells getting older. They can get in a normal way older, means they start to deteriorate faster when we are like 60 or 70 than if we are 20 years old. The metabolic structures in the cells called mitochondria, what are little fabrics, what are delivering energy and what let the cells replicate themselves, they getting damaged through influences from outside like toxins, from inside like viruses. And aging process starts with a deterioration of delivering energy, with a deterioration of the mitochondria. Then, when we talk about aging, we have to talk about abnormal aging of the cells. If there is a problem when the cells start to deteriorate because they're dying faster through toxins, viruses, etc. But at this time, the genes get damaged. The genes get switched on and off, what is called epigenetics, through a blockage of genetic material what has to do with detoxing fast enough over the liver, for example. When toxins are hanging in the cells, cells get damaged and they go down, so we age faster. Or an abnormal aging is when the cells get hurt and they deteriorate and they get destroyed very fast, so we can measure that in a building of cell trash, what is high in the cells, and we can measure that over the balance of abnormal dying cells and repairing cells. So we need a balance between cells getting repaired and cells are dying, what happens every day. So we can measure over mitochondrial functions how abnormal fast the cell is dying over inflammation, and we can measure on the other side over telomeres again, or over human growth hormone or some hormone systems, how fast can the cell repair. So we need a balance. The third abnormal damage in aging is if the cells mutating in a very bad way, going in the direction of cancer. So if cells are abnormally mutating, normally nature is eliminating them, so they go down, they get killed without building a disease. So if this process is disrupted because we have stress, we have corona in our life, we have viruses in our life, then they block the building of new cells, they block the human growth hormone, they block the hormones. All of this gets disturbed, and this can lead at the end of the day then into an abnormal growth of cells, what is called cancer at the end of the day, or it can lead to a point what is called apoptosis, cell death, that the cells can die too fast. And when organ systems or cells in organ systems dying too fast, we coming to diseases like diabetes, to diseases like liver diseases, fatty liver. So we can control these parts over specific stem cell markers to see how solid are the stem cells, what is good. Because when we know that stem cells are going down abnormally, we know what we need to do. We can take the same stem cells from the patient, called autologous stem cells. And this is a very important part. We can measure what is called clotogene. Clotogene is a gene complex in aging cells. When we can switch them on, we have a very, very good effect if cloto cells are switched on, clotogene switched on, that the cell itself can repair over the stem cells in a very good way. So there are different ways to block a cell from aging. There are different ways to detect what kind of aging process we have, because it's very important from the diagnostics. Aging starts on a cellular level where we look into genes, where we look into telomeres, where we look into mitochondria to detect what part of the cell is going down faster and what part is still stable. So tailor-made, we can make therapies there to stop the aging process.